At the beginning of meiosis, DNA strands thicken into chromosomes homologous or like chromosomes begin to approach each other. Homologous chromosomes pair to form bivalents. Centrioles divide and move to opposite poles of the cell. The bivalents duplicate to form tetrads or four chromatid groups. The nuclear membrane disintegrates and crossing over occurs. In metaphase 1, the tetrads line up at mid-cell. In anaphase 1, the tetrads separate and the paired chromatids move along the spindles to their centrioles. The cell membrane begins to constrict. In telophase 1, nuclear membranes close around the separated chromatids. The division ends and there are now two cells. Prophase 2 begins and the chromatids separate. In metaphase 2, the chromatids line up in mid-cell. And in anaphase 2, the now separated chromatids approach their poles and the cell membrane begins to constrict. Telophase 2 has been completed and there are now four cells, each with half the number of chromosomes of the parent cell. Before mitosis begins, each chromosome makes an exact duplicate of itself, and then the chromosomes thicken and then coil. Prophase begins and the centrioles, which have divided from asters, move apart. The membrane begins to disappear, and the centrioles and asters are at opposite poles. The nucleolus and the nuclear membrane have almost disappeared. The double chromosomes line up at mid-cell in metaphase. In anaphase, the centromeres split, half the chromosomes move to one pole and half to the other. The chromosomes have almost reached their poles and the cell membrane begins to pinch at the center. The cell membrane completes constriction in telophase. Nuclear membranes form around the separated chromosomes and mitosis is completed. There are two cells with the same structures and a number of chromosomes as the parent cell.